All right, well, welcome everybody. Thank you so much for joining us this evening. Those of you who don't know me, I suspect most of you do, but if you don't know me, I'm Jim Perkins, and I'm the director of the graduate program in medical illustration here at RIT. And we have seven of our second year graduate students who are going to be presenting their thesis work tonight. And we're going to have each student is going to take about 15 minutes. Um, I've allocated 2 full hours for tonight's session, so that means we probably have a time for just a couple of questions at the end of each student's presentation instead of trying to hold them all to the end. So let's go ahead and dive right in. Julia, you want to take over the screen and give us a quick introduction to yourself and take it away.
Yeah, sounds good. Just to make sure everyone can see this.
Looks good.
Awesome. I'm Julia, and my thesis project is called Explore the Human Eye, an Interactive Experience. So here's a little bit of scientific background. In 2019, a survey was done by the Harris Group and found out that majority of the adults that were surveyed were knowledgeable about eye health, but only a fifth of them are able to identify the three most common causes of blindness in the US, which is glaucoma, age-related macular degeneration, and diabetic retinopathy. And here are some statistics about vision impairment. So according to the CDC, over 14 million people in the US have vision impairment. So that can include like myopia um, and astigmatism and all that. About a million of them, or a little over a million of them have a form of blindness, and 6.3% of children under 18 are diagnosed with visual impairment. And one of the leading causes of blindness in the US and globally is cataracts, which over 20 million Americans over the age of 40 have, and almost 2 million Americans older than 40 are affected by age-related macular degeneration. So the reasoning for studying this topic, inspired by my own eye health, I realized I didn't know much about my eye and vision health as much as I should. Every time I went to the eye doctor, they asked me if I knew um, any changes in my eye or like if I understood anything that was happening, and I said no idea. Um, Last semester, I took interactive media with Craig Foster and learned about Sketchfab, which allows users to interact with 3D models that you create. And that kind of helped inspire the big part of this project. And the past 2 years, I experienced a lot of difficulty with Autodesk Maya. So I wanted to improve my skills by continuing to use Maya and try to implement it more in my work. So when you look up online interactive models of the eye or illustrations of the eye, you get a lot of illustrations and artwork, and almost all of them all vary in the anatomy of the eye, and there's not a lot of like consistent um, anatomical accuracy when it came to a lot of eye illustrations. So my content advisor and I went through and found some of the main interactive models that you can find on Google, like the first couple of searches, and pretty much analyzed every single model in detail to figure out which kind of specific anatomy worked well in those models, which ones didn't, and took all of that we analyzed into the model that I will be making. So the goals of this project is to create an accessible website that provides easy to understand information for patients and just everyday people. I wanted to create an interactive model of the eye that helps people understand their eyes better. That includes just a normal healthy eye, but also models of different eye conditions. So along with the different eye conditions, I wanted to highlight the anatomical changes that occur in them, and along with the interactive models to create downloadable content that patients and everyday people can download all the information that they learned and some of the static images of the model. So my content advisor is Jennifer Kresh, who is an ophthalmologist at the U of R Flom Eye Institute. And she sees patients all the time and kind of really helps me figure out what's the most important things to highlight in the project. And one of the biggest things that she taught me and told me about was what she looks for when she's examining patients' eyes and how to kind of implement that in my models. She also has a fine arts degree, which also helped and showed me a lot of the different drawings of the eyes she's done before. And so anytime I brought her a sketch or a model, she would give me pointers of what to make it better and what to do to help it be as accurate as I possibly can. I also want to thank Craig Foster, who pretty much also just helped me the entire time with modeling on Maya and all the troubleshooting on that program. And also for Jim for like helping me and Jen be able to get into the anatomy lab to look at and dissect actual cadaver eyes to just like look at the anatomy on those. So a lot of this project was a lot of like the different ways that I approached to creating these models. The first thing was understanding that the eye is extremely complicated. And so I did a lot of the basic research and trying to find as many visual references as I can. So Craig sent me a few textbook references for the anatomy of the eye. And on my most recent annual exam for my eyes, I talked to my eye doctor about this project. And he was like, here, take this model, take photos of it, take any photos in the office. So this was one of the models in there. And after reviewing this model and talking with Jennifer, a lot of it is not super accurate, but it gives you a pretty good overview of the eye. So the first step in creating a model on Maya is researching the measurements of the structure of the eye. The eye itself is not a perfect sphere. There's a lot of different variations in the diameters. So what I did was one of the references Craig gave me and then some of other surveys gave average measurements of the different diameters of all the different structures in the eye. And basically what I did was compare all of them and kind of find what's the similar variations in all of them. And basically the main basic model was formed from all these measurements. And that's a little screenshot on the corner from the first kind of iteration of it on Maya. So the other step was deciding what anatomy to include. So obviously the eye has a lot of very intricate structures. So meeting with Dr. Kresh, we kind of took a lot of time to figure out what's the most important structures to highlight in the model, especially for patient education. So we took out a lot of things that might have been too complicated for just everyday people to look at for the model. And also which eye conditions to look at. Obviously, there's also hundreds of different kinds of eye conditions that vary, and they all vary in how they affect others. So these are the main five that she believed was the best ones to focus on, and it's the ones that she sees the most with her patients every day. And the image on the side kind of just shows you some of the notes that I would take during our meetings and some of the things to highlight in the project. So here are some screenshots of the process of creating the model. So a lot of it was done in Maya and also Autodesk Mudbox for the painting. A lot of the painting was done through several uses of visual references and also some guidance of Jennifer. And so painting was done on Mudbox and everything else was done on Maya. So here is the final render of the normal healthy eye. This is done again in Maya, and this gives kind of a representation of the anatomical structure of the eye based on the measurements that were collected. Here is the inside of the eye, so users will be able to like kind of travel inside of the eye. That's a little painting inside of the retina. So once you travel into the eye model, you'll be able to see that. It's not completely done yet, but that's just like a basic painting texture placed on it. And then on the right side is the lens being held by the zonules and suspensory ligaments, and then the ciliary body around it. Here is the exact same model. So there'll be two main models. and this one's just basically just the model cut in half so that users will be able to see the different layers of the eye. Here is an example of creating different models for the different eye diseases. So this is just a screenshot of what it would be like for someone that has cataracts. So cataracts are caused by like the clouding of the lens in the eye, which is one of the leading causes of blindness in the US. So this kind of shows the difference between the two of like a normal healthy eyeball and a person that has cataracts. And this is an example of the model once it's published and embedded on Sketchfab and what users will be able to do with it. Each annotation will have a description about the function of those structures. And here is kind of like a layout of what the website's going to end up looking like. Currently, I am trying to use all different kinds of website building platforms to see which one's the most efficient and which one I like the most to creating this kind of experience. Overall, like users will be able to open up the model, the main model, and then have options of which conditions they want to look at, such as like the float, like looking at floaters, diabetes retinopathy, glaucoma, and all those other eye diseases, as well as they'll be able to interact with all those models, as well as download the information. And I'm trying to integrate more like interactive, engaging material in there to help everyday people and patients be able to learn with the health of their eyes and the anatomy of their eyes. And here is the project progress. Pretty much I have the research and the modeling of the eye models completed. The next couple of weeks, I'm going to focus on kind of designing that website to make it engaging and efficient and easy to navigate, and then finally publishing it. And that's it.
Thank you, Julia. That's awesome. It's a really nice model. I love seeing all of the research that you did, especially all those very detailed measurements about about the shape of, or the size of the different parts of the eye. Great job, by the way, sticking to your allotted time too. Kudos. So that means
Thank
we
you.
definitely have a minute or a couple minutes. If any folks have any questions, you could raise a hand or just shout it out. I see something in the chat. I don't know if I can bring that
Julia,
up.
I have a question.
Yeah.
Um, so I really love this. I think it's so good. And as a person who's been suffering from eye stuff since I was 11, like I still do not understand any of it. Um, my question has to do with like the eye that you are representing. Did you find in your research and in the choices you had to make that you were choosing like your eye would be a typical eye from like a healthy 20-year-old, or like if you had chose like a 50-year-old eye or like a baby eye, like are there age-related changes that are different just anatomically that are not pathological, but just like age related?
Yeah, so like molecular, like the age-related molecular degeneration is based on like when you get older, there's a lot of anatomical changes. So that kind of affects just how older people will be able to see. The model that I based it out of was just like very basic measurements that I found and pretty much just put it all together from that.
Okay, thanks.
Thanks, Sheila.
Heather, I see your hand up.
Hi, I'm here. Question for you, Julia. When you look, I love that you show the zonules all the way around the eye. I think that's something that's really hard to appreciate in 2D representations. Have you considered different colors or textures for those so they don't look quite like stitches?
Yeah, so also that's like big thank you to Craig for helping me figure that out. He really helped me a lot with the model. I am adjusting the colors for that because I don't like it to just be like the black like stitch like. So I will be changing that in the final model.
I think it looks great already, but
Thank
that was
you.
just the one thing that I thought. Great job.
Thank you.
Great. All right. Well, let's move along. If there are other questions, we can hold them to the end, but so we don't get behind. Um, Kirsten, you're up next.
Hey. Is it working?
Looks good.
Okay, great, thank you. So my name is Kirsten Santiago, and my thesis explores the olfactory anatomy and physiology of the dog and its application to medical detection dogs, volatile organic compounds, and medical detection screening. I'm having an issue with my presentation. So my chief advisor was Craig Foster, who basically held my hand through the entire animation portion of the project. And my content advisor was Dr. Otto from Penn Vet Working Dog Center. She is the director of the program, so she was incredibly knowledgeable and helpful, not only about the science, but also what would be good for other people to learn. I also received advising from Glenn, Jim, and Alan, who each helped guide me through the process at different points in time. And I would like to thank each and every one of them for the time they devoted to giving me feedback and helping me work through this project.
Thanks.
Is the next screen showing? Oh, okay. So my project motivations, anyone that knows me knows that I've always had a strong affinity for animals and a strong love for animals, specifically dogs. And I've always been interested in the intersection between humans and animals and how their relationship and interactions
Thanks.
work on a deeper level. I have also always been interested in animal behavior and how anatomy and physiology influences this behavior. And until last summer, I really didn't know that medical detection dogs in this context were a thing. And a lot of people I've spoken to also didn't know that. They knew about it in the context of medical alert dogs, but not in the detection sense. So I really wanted to create a visual information project that would help people understand this and engage with it more.
Thanks.
So, what are medical detection dogs? They are animals that have been trained specifically to detect scent markers, oftentimes associated with medical disease and disease biomarkers. These dogs have an extremely high abundance of olfactory receptor neurons and proteins, much more than humans, that make them extremely attuned to the scents around them. They can both detect and distinguish these smells at extremely low concentrations and at a much higher acuity than humans. So it's like a drop of something in a pool of water. And they can not only detect that the drop is there, but they can also detect what that drop is. And unlike humans that are generally dependent on their eyesight to navigate the world, dogs navigate the world through their nose. So, what are volatile organic compounds and what composes and creates scents? Odorants are made up of, like I just said, volatile organic compounds, which have high vapor pressure and low water solubility. And this essentially means that they easily evaporate under normal atmospheric conditions. So, as you're speaking, particles are leaving your mouth, and these particles could be considered volatile organic compounds. Biologically, they're released by plants, animals, bacteria, viruses, etc., due to metabolic cellular activity. these emitted VOCs can be biomarkers for various different diseases, including cancer, neurodegenerative diseases, infectious diseases, and non-transmissible bacterial and viral infections, but this needs a lot more research, especially if it's going to be addressed from a canine sense. And different combinations and concentrations of VOCs greatly contribute to scent composition, making individual scents highly variable. So every individual and species will have different scent combinations, so it makes it an extremely complex topic. So my introductory animation and website. My project deliverables include a website with a multimedia animation and an interactive learning module that includes multiple chapters and corresponding lessons that utilize 3D modeling and 2D illustration. Because I only have 15 minutes, I cannot show everything, but I will be playing the introductory animation and giving a brief walkthrough of the website. So my methods, I used the illustration components used Adobe Photoshop, Fresco, and Procreate, and the 3D modeling was done in Autodesk Maya and Mudbox. These animations were created in Autodesk Maya and Adobe After Effects, and my website was created with Squarespace. In order to make it interactive, I utilized an app that I recently found called Genially and Sketchfab, which basically allow you to create these complex interactive systems without the use of coding, which if you know coding, you know it's always nice to find a shortcut through that. And I was able to use iframe embed to bring these into my website. So here is my animation.
Have you ever considered how medical detection dogs can sniff out some of man's greatest predators? Like most animals, dogs have a split olfactory system, the main olfactory epithelium and the vomeronasal organ, which detects pheromones and low volatile substances. During sniffing, 12 to 13% of the airflow goes into the olfactory system, where odor molecules activate chemoreceptors that send signals to the brain. The remainder of the airflow enters the pharynx and lungs. Some of the scents that dogs sniff in can reveal hidden secrets to the human body and health. Volatile organic compounds are trace odorant gases that are produced during cellular metabolic processes. These VOCs often are biomarkers for certain infections and medical conditions such as infectious diseases, cancers, neurodegenerative diseases, and viral and bacterial infections. For humans, many of these scents fly under the radar, but not for dogs, because these scents are not only detectable, but also distinguishable. Dogs have an extremely high abundance of olfactory receptor neurons, which are activated when odorant molecules, such as VOCs, make contact with odorant receptor proteins on the receptor cell microvilli. This leads to a chain reaction, causing the olfactory neuron to send a signal that activates the olfactory bulb, notifying the brain about the stimuli. Have you ever?
And then here is my website. So when you first open it, you'll be on the landing page, and there will be the introductory animation that we just watched, as well as a brief interactive overview of the course content. I wanted to make the website extremely engaging, so I created multiple interactive photos and short frame animations. They will be able to view each chapter and lesson, as well as track their course progress through the progress bar percentage. Upon starting the course, they will be able to read about and interact with the microscopic anatomy of the dog nose and the olfactory system. And furthermore, when they hover over the icons, the corresponding anatomy labels will appear. The next chapter describes physiology, which has short animations that describe the airflow patterns and another interactive image that describes bilateral scent differentiation. Each chapter is followed by a short assessment, which just serves as a brief way to examine how well the information has been absorbed. Very low pressure. The section also has a corresponding lesson that describes physiology in which the viewer can examine, or this has a 3D model where you can scroll around and examine a neural cell, a neural olfactory cell, and you can basically turn it all around and look at all of the different components. There is also a corresponding physiology lesson where you can look at the different lock and key variations and what is lock and key and what's not lock and key. And you can also examine how signals are sent to the brain. After this, the viewer can develop a schematic understanding of volatile organic compounds and try an exploratory demonstration where you can identify a coffee bean. And this is connected to the biological detection, and you can gain a quick understanding of general diseases and the potential that these diseases can be detected in the laboratory setting. The last chapter describes the training methodology and tools that are used to train these dogs. And within a few weeks, I do plan to have an interactive game where you can essentially use the tools that you've learned about in the chapter in order to train your own pseudo detection dog. So to conclude, I had a really amazing time exploring the complexities that make the canines capable of detecting these scents with such a high acuity. My hope is that further artwork and exposure for the topic will engage more people, creating the need for further research to streamline these detection studies and training methodologies to bring their potential uses to fruition, because right now the research is just not enough. And as the semester comes to a close, I would like to complete the training game and add a bonus chapter that describes the characteristics that define a successful medical detection dog. So just like humans, some dogs have greater capabilities to do certain actions than others. And I once again would like to thank my thesis advisors, the program faculty, and my cohort for supporting me through this process. Thank you. Please let me know if you have any questions.
Awesome, Kirsten. Yeah. And once again, great job staying within your time. Yeah, we definitely have time. If anyone has a quick question or two, again, you can raise your hand or just shout it out. I don't hear anything, so I'm going to jump in real quick. But if anybody else has something, feel free. I've heard about these dogs, but frankly, I know very little about them. What are some of the illnesses that they're able to detect currently, or what sorts of things are they looking into for them to be able to detect with their olfactory organ?
There's quite a few cancers that are being examined specifically. So lung cancer, ovarian cancer, and prostate cancer are being researched, but it's very small studies. So it's not necessarily a situation where you can say, oh, like these dogs can detect them, so I can just go find one and they'll be able to do it. Like it's very much in research and it's in progress, and they may eventually be able to find a way to implement this. There's also neurodegenerative diseases like Parkinson's. I know Parkinson's has very specific scent markers. Um, COVID has been examined a lot with this. Trying to think. Malaria, I believe I've read about. And there's also like specific bacterial infections. So I think staph infections.
Hmm.
A lot, but
Yeah.
It's a
Yeah,
lot, but still needs a lot of research.
Sheila, you got a question?
Yeah, I love this presentation. This was so
Thank you.
amazing and yeah, great work, beautiful animations, everything. Um, I think it would maybe be a cool thing to consider adding like a donate to research in this area button to your website because people will, I mean, I'm ready to like give you some money for this research. I think it's really interesting, and I know that's a a good way to maybe raise some small funds for the people who are doing this.
It's a great idea. I didn't even think about that. So
Dave.
Uh, Kirsten, I did my PhD on nasal chemical senses, and if you want to pick my brain, I have some suggestions for you, and also about the volatile diseases that dog can detect. I also used to clicker train dogs, so so we should talk.
yeah, that would be great. Thank you.
Quick question. Do you know if this is the same mechanism by which they say dogs and cats can detect death? Is it the same volatile chemicals situation, or do you know if that's a different thing that they're scenting?
I don't know specifically. I would presume that it has something to do with that. I'm sure there are certain scent markers associated with like death and decomposition, but I couldn't tell you for sure.
It may be an interesting thing to look into because I know a lot of people who ask about that before they ask about disease detection. So it may be a way to like segue.
Yeah,
It's really interesting though. I love your illustrations.
thank you.
All right. If anyone else has questions, let's hold them to the end. So let keep moving ahead. Krista.
Awesome. Let me share my screen and get this set up. Okay. So my name is Krista. I'm an MFA Medical Illustration graduate candidate. And today I'm going to be talking about a novel approach to 3D printing nasal septum scaffolding with the associated 3D modeled assets for use in a virtual surgical environment. So a little bit about me. I grew up in Omaha, Nebraska. I got my bachelor's in neuroscience from the University of Nebraska at Omaha in 2014. After graduation, I worked in a neuroscience lab and I continued to take digital media classes to learn art, digital art. And then in 2018, I moved to Georgia and continued to take courses in anatomy and physiology. When I moved to Athens in 2020, I took some scientific illustration classes at the University of Georgia before moving here to Rochester. And here I am at Imagine RIT last year at the prosthetics booth, making a mess. So a quick overview. I'm going to go over the objectives, the background science, some design concepts, the printing process, some concept sketches that I created, 3D assets, and then future directions before going to acknowledgements. But I did want to acknowledge my content advisor, Dr. Mark Shigowitz. He provided funding for this project for the 3D print portion. And then I also wanted to give a shout out to the tissue engineer, Dr. Karen Wertz-Kozak, Dr. Jade Myers, and then Jillian Silva for the support. So some objectives. The first objective for the team was to create and design a print that was a biocompatible 3D scaffold. This would be a prototype that could be biointegrated into native bone tissue and then support the growth and proliferation of a hyaline cartilage. For my portion, I wanted to create a 3D patient bust and surgical tools to be used in a virtual reality environment to help medical students practice the implantation of the novel scaffold. So a little bit of background on the nasal septum anatomy. The nasal septum is a four-sided structure made of bone and a hyaline cartilage. It sits medially within the nasal cavity. It's responsible for dividing the nose into two lateral chambers. It attaches superiorly to the perpendicular plate of the ethmoid and inferiorly to the maxillary crest. and then it protrudes anteriorly to the nasal tip and posteriorly to the nasopharynx. The hyaline cartilage portion of the septum consists of chondrocytes and lacunae, which is shown in the purple box. These cells are isolated within an extracellular matrix of collagens and proteoglycans, and these are fed by the branches of the maxillary artery. The role of the nasal septum is to provide structural support for the nose. It's also covered in a mucous membrane, which warms the incoming air before it reaches the lungs. The hyaline cartilage portion is avascular, which means it is without blood vessels. So this part is delicate and susceptible to damage, and it's a common site for congenital defects. So for the material selection for the 3D printed scaffold, oh, I think I skipped one. Oh, here we go. So some common nasal deformities and damage that can occur. Nasal perforations, which are holes in the nasal septum. Typically, this can occur for various reasons like surgical mishaps, sometimes in the case of like rhinoplasty, a deviated septum, which is a congenital defect, and it occurs in approximately 20% of all newborns. Trauma can occur, so this can happen during military combat or just surgical trauma. Cancer. So nasal tissue is actually the most common site for facial skin cancer. So nasal cartilage is often damaged when the tumor is resected. Damage during drug abuse, so those that use cocaine and abuse nasal sprays, these actually act as vasoconstrictors, so they can restrict the delivery of nutrients to the nasal tissue and cause cell death. And then the effects of this are discomfort, trouble breathing, dryness, bleeding, crusting, and just overall reduced quality of life. So some of the current repair approaches, there are nasal prosthesis. This is a non-surgical approach, and this is just a covering over typically nasal perforations that are on the smaller side. And this is a good option for patients that are not good candidates for surgery. Septoplasty is just the removal of damaged tissue, and then allografts and tissue transplants. So these can vary widely based on the donor tissue. So currently there is no standard method of treatment for nasal cartilage repair. Okay, so for the material selection, we were looking at certain properties of the print material. This included biocompatibility, cost effectiveness, is it biointegratable, structural integrity, and then high print resolution. We wanted to be able to control the pores of the print so that we can maximize cell penetration, growth, and differentiation. So my initial research focused on alginate. This is a biocompatible material. It's readily available and cost effective. And it can be prepared so that stem cells are influenced to differentiate into chondrocytes. The downside of this, however, was that hydrogels require stem cells to be mixed in with them before printing, and I do not have access to stem cells. So we did not go that route. However, that will be an important second step of this process as the research continues. So upon further research, we found that hydroxyapatite was a good candidate. It's bioceramic, it's biointegratable. You can adjust the pore size. Studies show it has successful osteoconductivity, and it can integrate into native bone tissue. It allows for proper printing of pore size, so we can adjust that as needed. And studies show that there is a high cell proliferation rate with stem cell seeding. So we ordered this material that you see on the right from a company called Lithos. And along with producing this material, they also have bioceramic printers that happen to be located here on campus. So it worked out. So for the scaffold design process, one of the research questions that we had was, how will the geometry of the scaffold affect the growth and proliferation of these cells? So these scaffolds are designed to be printed and sent off for in vitro cell testing, which we wanted to create several lattice variations. So these variations were created in NTOP by the industrial design student Jillian Silva. And the overall shape of the geometry was kept simple to ensure proper printing, but in the future, the intention is that the overall structure would take more of the shape of the nasal septum. So we use current research to understand the ideal pore size for cell growth, so the measurements of the pores in these particular scaffolds reflect that research. For the printing process, we use lithography ceramic manufacturing, or LCM. This uses the hydroxyapatite slurry that is dispensed into the vat, which is shown in number 2. The vat will rotate and the slurry will coat the vat. Then the building platform, seen at number 1, will be loaded upside down. It will come down into the vat, and then the LED light will shine up through the optical system and cure the hydroxyapatite onto the building platform. So on the right, you'll see the printer screen that we used with the printer here on campus, and you'll see some of the lattices that were created, loaded into that screen. So here's a quick GIF of the print process. You'll see the vat spinning with the hydroxyapatite slurry coating the vat. And then the build plate will come down, the vat will tilt back up, and that will be that layer will be cured to the plate. So this process continues until the entire thing is built. In the image on the right, you'll see that once the build plate comes out, this is how it looks. This is the 3 different lattice variations, and it still has some of the excess slurry on it. So this is how it looks before it's cleaned off to reveal the pores. So here are the print results showing the different lattice variations that were created. As you can see, it does have good print resolution for what we needed. There were a couple bumps in the road where the printer settings were not quite dialed in yet, so it took a couple tries, but it didn't take long to get some successful prints like the ones shown here. So for the visual portion, the 3D assets that I created, I wanted to create surgical tools. So I did this in Autodesk Maya, and I textured and added materials in Adobe Substance Painter. Then for the bust, I used photogrammetry. I used an app called Polycam, and I took the file that was created in this app into ZBrush to clean up the geometry, do some sculpting, and then I used Polypaint to paint directly on the model. So here are the initial sketches that I created to show the surgical tools and how it might look in the VR environment with the patient bust. And the rotating GIF is a 3D scaffold concept that the industrial design student had designed just so I could add something in there for imagery. We're not entirely sure how it's going to look in the end, so I wanted to add something. For the surgical tools, I wanted to model some bite liner retractors, so I started with some reference photos that I took, and I used a free image plane with that image. I modeled on top of that in Autodesk Maya, and then once I modeled one side of the retractors, I just mirrored it, and then I modeled the locking mechanism. So then on the right, you'll see I UV mapped each piece in Maya so that when I brought them into Substance Painter, the software could read the UV map in a 2D space, and I would be able to add texture onto it before exporting. So I use the same modeling process with the scalpel. I modeled on top of a free image plane. This is a number 15 blade with a number 7 Bard Parker scalpel handle, which is typically used in septoplasty surgery. And you can see the geometry that I modeled for each part, and then the UV maps on the right that I created in order to add the materials and textures in Substance Painter. So these are the final renderings of the scalpel blade and handle. This includes the textures and material. For the bust model, I used photogrammetry, so I used my own head as a model, and then with the Polycam app, I had Craig take several pictures of my head, and the app will take those pictures and splice them into a 3D model, which can be exported in various formats. And as you can see, it comes with triangular geometry, which is not ideal to sculpt on. So I took that into ZBrush and used ZRemesher to convert it into quads to make it easier to sculpt. Then with ZRemesher, I was able to reduce the poly count, and then you'll see the model on the right. I was able to sculpt in some wrinkles, eyebrows, various details. For the next part, I created a surgical cap model. So I sculpted this from a basic sphere in ZBrush. I used the cloth pull and cloth nudge brushes to kind of pull the geometry around to give it a little bit more of a natural cloth look. And then I applied a blue fill color. So for the skin colors, I used poly paint with the spotlight tool. The spotlight tool allowed me to color pick some of the colors off of my photogrammetry bus, so I was able to paint those directly on the model. So here is sort of a mock-up. I added a table and a surgical drape. So for the drape, I used a flat plane in ZBrush, and I used ZBrush Dynamics with gravity, and the drape would fall onto the model to give it a more natural looking cloth effect. So again, here's a quick mock-up of what I would be placing into the VR environment. So some future directions. The 3D scaffold prints are going to be sent to the lab of my advisor, Dr. Shikowitz, for in vitro cell testing to test for cell viability. He plans to write an NIH grant, which can support further research, and hopefully in the next phase of the print, which would include the hydrogels mixed with the stem cells so that they're geared toward differentiating into cartilage cells. And then for my portion, I plan to finish modeling the tools, the forceps, needle holders, and surgical needle. I want to create a blend shape for the incision opening to have it open for those that are interacting with it, and then add these assets to a VR operating room environment with Unreal Engine and create blueprints for users to interact with the surgical tools and the patient busts. So I just wanted to say thank you so much to my academic advisors, Jim Perkins, Craig Foster, Glenn Hintz, and everyone in their research and development and design portion of the 3D print. And then again, thank you to my content advisor, Dr. Mark Shikowitz, and everyone that provided academic support, my peers and friends and family. Thank you so much.
Thank you, Krista. Great presentation. Wouldn't surprise me if a lot of people have some good questions. I think we have maybe time for one quick question, just because we're a little behind because of the technical issues.
Sorry.
I know. It's funny, it worked great at the beginning, when we were testing it. And then any quick raised hands or got some kudos? Good job. Right. If I'm not hearing any quick questions, I don't see any hands up. Why don't we go ahead and move on to Sophia?
Yeah, that's good. Okay, let me share my screen. Oh. Okay, so hello, my name is Sophia Chen, and my thesis is on the retinal abnormalities observed in patients with schizophrenia. First of all, I would like to give credits to everyone that has assisted me in this project, which includes my thesis advisors, Jim and Craig, and my content advisor, Dr. Steven Silverstein, at the Center for Retina and Brain and the Schizophrenia Treatment and Research Lab at the University of Rochester. Also, thank you to Bailey Maikitak, who's a graduate of last year. If you are here, thank you for letting me borrow your 3D model of the brain. Retina's involvement in schizophrenia is a very niche and under-researched area. So the purpose of this project is to raise awareness of this field and to inspire additional research initiatives. Ultimately, it aims to advocate for retinal exams as a monitoring tool in psychiatric disorders for potentially improved patient outcome. To do this, I planned on making a 3D animation explaining the basics of the retina and its associations with schizophrenia, and a website that further details the findings.
To give a bit of background information on the science, schizophrenia is a psychiatric disorder that involves delusions, hallucinations, and disordered thinking. Historically, it was understood as a disorder of the brain, but now it's being recognized more as a systemic illness, especially since there have been changes observed in the retina of schizophrenia patients. So I will further describe the retina in my actual animation, but to put it briefly, the retina is a layer of neurons in the back of the eye which detects light and sends signals to the brain. And why should we study the retina? Well, embryologically, the retina arises from the same neural tissue as the forebrain. As we can see from the image on the right, it is derived from the optic vesicles, which are out pockets of the developing forebrain. The retina also uses the same neurotransmitter as the brain, which is glutamate. And the ganglion cells in the retina directly form the second cranial nerve, which synapses with the lateral geniculate nucleus of the brain. And for this reason, the retina is actually considered to be part of the central nervous system. And it can be visualized inexpensively and non-invasively compared to the rest of the CNS. In my thesis, I focus on the 4 significant retinal findings in patients with schizophrenia, which are thinning of the retinal nerve fiber layer and thinning of the macula and reduced microvasculature and abnormal electroretinogram. So the retinal nerve fiber layer, or the RNFL, is the layer of the retina that forms the second cranial nerve. And at the Crab Lab, they measure the RNFL thickness with optical coherence tomography, which basically shines near infrared beams into your eye and then measures the amount of light reflected back through your retina. And the macula is a small region in the center of the retina responsible for central vision, and its thickness can also be measured with OCT. This image on the right is a picture of the microvasculature, and as we can see, there's thinning in the schizophrenia one. At the Crab Lab, this is measured with OCT angiography, which is done at the same OCT machine. The OCT angiography visualizes the blood vessels by detecting the motion of blood flow, which is non-contact and a lot less invasive than the traditional angiography methods that require dye injections. And at the lab, retinal electrical signals are measured through flash ERG, which produces the image over here. The alpha wave of the ERG reflects the hyperpolarization of photoreceptors, and the beta wave reflects the depolarization of Mueller and bipolar cells. The photonegative response reflects activity of regional ganglion cells. And to perform this, they place an electrode strip on your bottom eyelid and flashlights into your eye through this handheld device. So to better understand the procedure and findings, I paid a visit to the Crab Lab myself and had a retinal test done. And this is me getting the ERG done, and these are the results of some of the results I got from that day. I'm not exactly an expert in interpreting these data, so I can only hope that everything looks normal. To plan for my animation, I first wrote an AV script, which corresponds each line of the narration to what happens on screen. Once everything got approved, I made a storyboard and recorded my narration at the soundproof narration room at RIT. To create my 3D models and animation, I used Maya, ZBrush, and Substance Painter. So these are all the types of neurons in the retina. And I made them all in Maya. The cell bodies are created with a primitive polygon, and the dendrites or axons are created through extruding one of the faces on a curve. For the texture, I was really interested in having a translucent sort of gummy bear texture. So I used transmission and applied a noise bump map to create the render that you see over here on the right. So for the half eye, which I used in the animation, I found a reference containing accurate dimensions of the eye. And I used the CV curve tool in Maya to trace the outline. Afterwards, I used the surface revolve tool to create the half sphere. And I repeated this process with every layer inside the eye. The full eye was modeled in Maya and then brought to ZBrush for sculpting. I found a stock image of the iris from the internet, and then I imported it into ZBrush and used the spotlight tool to transfer the color and texture onto my model. I also use Substance Painter to texture a lot of my models, and these are just some examples of the texture maps that I have exported. For the animation itself, I used a lot of dynamics in Maya, including mesh network and particle simulation. I used mesh for many different parts, such as constructing this 3D retina. I created 4 separate meshes for the different type of cells in the retinal layers, and I used random node to distribute them a bit more organically, and I used the ID node for the photoreceptors so that there will be distinctions between the rod and cones. I also use the N-particle system for simulating the neurotransmitter glutamate. In my background image of the presentation, like in all of the background images, I wanted glutamate to be released from the photoreceptor onto the dendrites of a bipolar cell. So I drew a CV curve and used the volume axis field option and played around with these attributes to control the speed and amount of particles emitted. So without further ado, this is my final presentation.
Schizophrenia is a severe psychiatric disorder that affects about 1% of the population. Symptoms of schizophrenia typically include delusions, disrupted thought process, auditory and visual hallucinations. In addition, 60% of patients report visual distortions such as impaired perceptions of brightness, depth, and color. While these symptoms are typically attributed to abnormalities within the brain, studies suggest that the onset of schizophrenia is associated with changes in the eye, more specifically, the retina. The retina is a web of neurons in the back of the eye which captures light and sends information to the brain. It contains 10 distinct layers: the retinal pigment epithelium, photoreceptor layer, external limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, retinal nerve fiber layer, and the internal limiting membrane. It is formed by three main cell types: rod and cone photoreceptors, bipolar cells, and ganglion cells. Other neurons in the retina include Mueller cells, a type of glial cell, horizontal and amacrine cells, which are interneurons in the OPL and INL, respectively. Although located in the very back, photoreceptors are the first to respond to light. In complete darkness, they depolarize and release the neurotransmitter glutamate onto the dendrites of off bipolar cells, which inhibit the activation of retinal ganglion cells, and no light is perceived. When light signals are present, photoreceptors hyperpolarize, which results in glutamate being released to on bipolar cells, which activate retinal ganglion cells, whose electric signals are communicated via their axons that travel along the vitreoretinal surface of the eye. These output axons form the retinal nerve fiber layer, which is separated from the vitreous humor of the eye by the internal limiting membrane. The axons in the RNFL bundle together and form the second cranial nerve, the optic nerve. It exits the eye through the optic disc and eventually synapses with the brain. The retina receives blood from the central retinal artery and posterior ciliary arteries. The posterior ciliary arteries reside behind the photoreceptors on the choroid. The central retinal artery enters the retina through the optic disc and branches into arterioles that form three retinal vasculature layers: the superficial, intermediate, and deep plexus. Other significant structures include the macula, a circular spot about 5.5 millimeters in diameter in the center of the retina. It has a high density of cones and therefore is responsible for the sharp color perception of the central vision. The fovea is a small depression in the macula and is known to be avascular of the central retinal artery circulation, an area termed the foveal avascular zone. Due to the retina's involvement in the optic nerve and its close connection to the brain, it is considered to be part of the central nervous system and often referred to as the window to the brain. Multiple retinal changes have been observed in patients with schizophrenia, such as thinning of the macula and the RNFL, reduced microvasculature, and abnormal electric signals. Although it is still unclear whether changes in the eye directly contribute to the visual symptoms, these findings suggest that retinal exams can be adopted as a cheaper and non-invasive tool for the monitoring of progressive central nervous system atrophy in schizophrenia.
So after that, my animation is finished. Right now, I'm working with the UX and UI person at the University of Rochester to hopefully have a page created on the Crab Lab website that will have my video embedded and a few more paragraphs explaining the findings that I talked about earlier in the presentation. And that's the plan. Thank you for your time.
Thank you, Sophia. That very impressive modeling. That model of the retina is incredibly detailed and beautifully done. Time for a quick question before we move on. We're not too far behind.
I have a quick question.
Okay.
Sophia, do you know if there's less glutamate in the retina than like there is in the brain for a lot of schizophrenics? So that's one of the characteristics of the schizophrenic brain, is just having a little less glutamate. So I was wondering if that was true in the retina as well.
Yeah, very interesting question. I actually haven't looked into the amount of glutamate in the retina, but I will definitely do more research on that. Thank you.
Yep. Thanks.
All right. Well, let's go ahead and move on. And again, if there are other questions that people want to hold to the end, we should have plenty of time at the end. So Winnie, would you like to take over?
You guys see that?
Yep.
Okay. So hello. I'm Winnie, and my thesis is on horizontal gene transfer in this area. So a little bit about me. So I grew up in Oakland, Maryland, and then I did my undergraduates in cell biology and genetics at the University of Maryland College Park. And right now, I'm at RIT studying medical illustration. So I'll start off with scientific background. So antibiotics are a really special class of drugs that help prevent bacterial infections by either killing the bacteria or hindering their growth and replication. The first antibiotic was discovered in 1928, penicillin, by Sir Alexander Fleming. And since antibiotic discovery, we have been able to treat bacterial infections efficiently. It also enabled many modern medical procedures such as organ transplants, open surgeries, and chemotherapy. And then it also extended average human life expectancy by 23 years. So antibiotics work in four main mechanisms. They either inhibit cell wall synthesis, they can bind to bacterial ribosome and inhibit protein synthesis, and they can also inhibit DNA and RNA polymerase and inhibit nucleic acid synthesis, or they can also bind to metabolic enzymes and inhibit metabolic pathways. But unfortunately, shortly after antibiotics were put into clinical use, antibiotic resistance was also observed. For example, tetracycline was introduced in 1959, but then in 1950, but then in 1959, tetracycline resistant Shigella was identified. And then just 2 years after the introduction of methicillin, MRSA was identified in 1962. And the appearance and spread of these resistant strains are a great threat to public health and safety. According to CDC estimate, 2.8 million antibiotic-resistant bacterial and fungal infection cases are observed in the US per year. And then of those cases, 35,000 will result in death. So how do bacteria render these antibiotics inefficient? So they can either do that by limiting the drug intake. That's more of a characteristic of gram-negative bacteria since they don't have two lipid bilayers. Some bacteria have developed efflux pumps to basically pump the drugs back out of the cell before they do any damage. And some bacteria also develop enzymes that will modify or destroy the antibiotic, and then others have gained mutations to the antibiotic targets. So the antibiotics can no longer bind efficiently to them. And of these four methods, the latter three can be gained through a few genes, which you can get through horizontal gene transfer. Okay, so the bacteria I'm going to focus on is Neisseria. Neisseria is a genus made up of several closely related gram-negative Diplococcus bacteria. And of the human-associated Neisseria species, only 2 species cause disease regularly. Neisseria meningitis, which is found in the nasal pharynx and is an opportunistic pathogen that causes meningococcal disease and bacterial meningitis. Neisseria gonorrhea is always considered pathogenic, and it's found in the nasal pharynx, urogenital tracts, and the rectum. It causes the STD gonorrhea in humans, and half of all gonorrhea infections are resistant to at least one antibiotic. Okay, and horizontal gene transfer. So there are 3 main categories of horizontal gene transfer. Transformation is when living bacteria actively take up free DNA present in the environment from dead organisms. Conjugation is the one-way transformation of DNA from one bacterial cell to another. And transduction is the spread of DNA between bacteria by bacteriophage. And when I was researching the topic, for horizontal gene transfer, I was usually just able to find still illustrations or really simple 2D animations. So for my thesis, my goal was to create a 3D animation that explains how horizontal gene transfer occurs in this area, focusing mostly on transformation and conjugation, since those are what scientists study the most. Okay. So the programs I use, I chose Blender as my main 3D modeling program, and I also use Illustrator, After Effects, and Chimera. And I also utilize online data banks such as Pumpkin and Protein Database. So to start off, I start off by writing up a script after I did all the research. I'm not going to read this off. You guys can hear it later in the animation. And then actually started next with the modeling process after I figured out all the characters I need. So most of the modeling is done with some pretty simple geometric shapes. And then I was also able to get the molecular structures of the antibiotics off of Pumpkin and getting them into Chimera and then exporting them to Blender. And then also, of course, model the Neisseria bacteria itself. And then put materials on everything. Okay, so I use a lot of geometry nodes in my modeling process. For example, this gram-negative cell wall, which you can tell by the double lipid bilayer and then the layer of peptidoglycan in between. This is actually just a plane with a geometry node applied on. Here's everything in the nodes. And the benefit of modeling a geometry node is that it's really procedural. So I can easily edit the thickness of this cell wall by just changing a few numerical values. And you can also key and animate geometry nodes. Another example of geometry node is modeling the DNA. So this DNA here is actually just a geometry node applied on a curve. And then the good part about this is that you can get a longer strand of DNA by just simply extruding the curve. It won't descale and distort the DNA at all. Okay. And then I also use a free Blender add-on called Molecular Nodes in this project. So this highlighted structure here is the type 4 pilus of Neisseria. And I retrieved that data from Protein Data Bank. And you can, with this add-on, you can directly go into Blender, type in the special 4-digit code for it, and it'll automatically grab it for you. And then you can edit the style of the protein model directly in Blender and also give them different colors. It just makes it really easy for modeling, so you don't have to jump between Blender and Chimera when you do this. Okay, so my next step was storyboarding. So after I got all my models done, I did some still image renders, brought them into Illustrator, add labels, and decide the layout I like, and then put them together in Google Slides with the narration to do the storyboarding. And then I went back and start keying stuff in Blender for the animation. So there was a bunch of simple animations such as keying the location and the rotation of objects, and also keying values on the geometry nodes. And I just want to show you this animation effect I was able to achieve in Blender using a geometry node. Basically, this was a dissolve effect. So whenever cells that have this geometry node applied comes in contact with this empty here, when the antibiotic is raining down, the cell will start to dissolve and release particles. And another interesting animation task I figured out was how to animate cell division with this modifier in Blender called a mirror modifier, which I basically reference each cell to an empty and then move the cell farther away from the empty, and the empty will act as a mirror, and it will give you the illusion of the cell dividing. And then finally, after everything was completely rendered out of Blender, I brought them into After Effects for the final compositing. And then here I will show you guys the final product.
The average human body carries as many bacterial cells as human cells. The majority of these bacteria are commensals or harmless bacteria that make up your personal microbiome. However, the rampant use of antibiotics in modern society is altering the composition of the bacterial population by increasing the frequency of antibiotic-resistant traits in both target pathogenic species and non-targeted commensal species. The problem becomes even greater for some commensal bacteria, such as Neisseria, which frequently pass genetic information to their pathogenic relatives through horizontal gene transfer. Unlike vertical gene transfer, where genetic information is passed down vertically from parent to offspring, horizontal gene transfer occurs between unrelated individuals or even across different species. This allows the recipient bacteria to acquire traits not possessed by its predecessors. There are three main methods of horizontal gene transfer: transformation, transduction, and conjugation. Of the three different mechanisms, transformation and conjugation are the main mechanisms observed in Neisseria. Transformation occurs when living bacteria actively take in free or naked DNA present in the environment. For example, Neisseria uses its type 4 pilus to scavenge DNA from their surroundings. Bacteria capable of transformation are considered to be competent, where their cell membrane contains protein channels that allow DNA to flow into the cytoplasm, aided by competence protein. Conjugation is the one-way transmission of DNA in the form of plasmids, which are small circular rings of DNA capable of replicating independently from the bacterial chromosome. During conjugation, the donor and recipient bacteria establishes cell-to-cell contact through the sex pilus, through which a single-strand replication of the plasmid moves from the donor to the recipient cell. The machinery needed for conjugation in Neisseria is encoded in the P-conj plasmid, which also encodes for tetracycline resistance. Once Neisseria acquires P-conj, it can spread additional antibiotic resistance traits through smaller non-conjugative plasmids, such as PBLA, which encodes for beta-lactam resistance. Given the commensal Neisseria's ability to spread genetic information through horizontal gene transfer, it is important for all commensal bacteria to be considered a potential reservoir for antibiotic-resistant traits for more virulent bacteria. Therefore, continued uncontrolled use of antibiotics aided by horizontal gene transfer will result in the rapid rise of multi-drug resistant bacteria.
And I would like to thank everyone who's on my thesis advisor, especially Craig for keeping me on track with his animation class, and then also my scientific contact advisor, Dr. Krista Wadsworth, who is a evolutionary biologist at RIT who studies antimicrobial resistant in bacteria. She also has a special focus on the Neisseria bacteria. And that's it.
Great. Thank you, Winnie. Awesome. Terrific animation. And any quick questions for Winnie? And feel free to put up your hand or just shout out. All right, I'm not hearing anything at the moment, but so just in the interest of time, let's go ahead and move on. But again, if you have questions for Winnie, feel free to save them to the end. So next up, Harmony. You want to take over?
Yeah. Can you see my screen?
Yes, I can see your screen. Can you get it to full screen mode?
Yeah.
Here we go.
Hi everyone, I'm Harmony Wong, and today I'm going to present my thesis, which is a 3D infographic animating series explaining the mechanism of disease of ventricular tachycardia after depolarization subtypes and triggered activity. Before I begin, I'd like to give a quick overview of my presentation. I'll introduce a bit about myself, also acknowledge my content and faculty advisors, and then I'll dive into the project background, concept, the objective and goals, materials and methods, my process of work, my final animation sequence, and also finally some of the future directions. So I'm from Hong Kong, and before I came to RIT for the medical illustration graduate program, I got my Bachelor of Science degree in biochemistry at the Chinese University of Hong Kong. I'd like to acknowledge my content advisor, Dr. Bruce Lerman. He's a professor of medicine and pediatrics from Weill Cornell Medical College. He is also former chief of the Division of Cardiology and director of the Electrophysiology Laboratory at Weill Cornell and the New York Presbyterian Hospital. His expertise is in cardiac electrophysiology, especially ventricular tachycardias. I'd also like to give thanks to my two faculty advisors, Jim and Craig, and also Glenn and Ellen, who have been giving me a lot of support along the way. Also, special thanks to my dad, Kevin, my family, my classmates, Brianna, Julia, Kirsten, Krista, Sophia, and Winnie, and my friends. I'd also like to thanks for Saline Trust for providing my research grants. So what is ventricular tachycardia, or in short term VT? Ventricular tachycardia is a type of abnormal heart rhythm or arrhythmia that occurs at the heart's lowest chambers, the ventricles. The most common VT is caused by structural heart disease. For example, a patient got a heart attack and lost some of the heart muscle cells, which disrupt the normal regular electrical signals that flow through the heart, leading to a mechanism called re-entry. There are two more mechanisms of arrhythmias, including enhanced automaticity and triggered activity. And we are going to focus on triggered activity to explore how an abnormal impulse formation would cause irregular heartbeat without a clearly defined reason, which is called idiopathic VT. To understand the disease, it's always better to know what is normal. Excitation contraction coupling is a process where the electrical signals or impulse triggered muscle contraction. Here's a diagram showing the normal mechanism of the beginning of a heartbeat. Sodium ions trigger the calcium ions to influx into the cell, which causes the increase in intracellular calcium that depolarizes the heart muscle cell. Calcium binds to troponin C on the muscle fibers, causing it to contract. When every cardiac muscle cell contracts in a synchronized fashion, the heart pumps. In an abnormal impulse formation condition, for example, overstimulation of the beta-adrenergic receptor, it will lead to overactivation of downstream proteins and trigger more calcium ions to flow into the cell in the easy coupling process. Eventually, it will generate a current outside of the normal beat. And you can see the little bump on the action potential and further become a triggered activity, which is an extra beat. This is called delayed after depolarizations, DADs, because it occurs at phase 4 of the action potential. There is one more mechanism that occurs earlier in phase 2 or 3, which is called early after depolarizations, the EADs. In this diagram, you can see the potassium ion channels are important to the repolarization, helping the heart to return to its resting potential by expelling the potassium ions out of the cell. When there are mutations of the potassium channels, outward potassium current will decrease, which also means that the voltage is high up. The problem of this is that the action potential will prolong, which will lead to long QT syndrome. And it will also reactivate the calcium channels to let more calcium ions flow in and trigger a current that eventually will become an extra beat. As you hear me explain the mechanism of disease, you can see that I often jump in between the diagrams, the action potential and the ECG, and also the ion channels. VT could appear in many different ECG patterns, and every ion currents have their own time frame. So to understand the whole concept of arrhythmias and ECG, it is important to show the relationship between the 2D graphs and the 3D world. I approach by creating a 3D animation series, and it consists of part 1 and part 2, with the part 1 explaining the normal EC coupling and the second part explaining the VT mechanisms. My objectives are to patient and educated audience. They will recognize and understand the concept of cardiac conduction system. And for medical professionals and researchers, they will clarify ECG misconceptions, understand and relate the molecular mechanisms of arrhythmogenesis, such as in long QT syndromes. And for biomedical communicators, instead of hand-drawing ECG patterns, I found a way to import the ECG data to Houdini and to generate procedural data visualization and animation. It helps me to visualize so many different VT ECG patterns and make it more accurate. And I hope the workflow could encourage fellow medical animators in producing data-driven graphics and animations. For materials and methods, I went through research papers, anatomical references, and met with my advisor before writing a script. I also researched the relevant protein structures from Protein Data Bank and their interaction with other molecules. For the ECG data, I extracted the data file from one of the arrhythmia databases and went through some data processing and cleaning up. In the pre-production stage, the script and storyboard are developed and referenced with slides and images. So you can see there are sketches, animatics, and concept art in the process. For the modeling, texturing, sculpting, lighting stage, I model the heart, the cardiomyocytes, and animate the molecules using Cinema 4D, ZBrush, Substance Painter, and Redshift. A low poly model was made and then sculpted with details. Texture maps, including bump maps, color maps, and displacement maps, are made in Substance Painter and then applied to the models. And then after that, I animate the scene and put in particle simulations to mimic the ion flows in Cinema 4D. Fields and particle emitters are used to control the trajectory of the particles, and time constraint, friction, and turbulence are used to give randomness, time control, and particle behaviors of the ions. And here, for the data-driven visualization, after cleaning the data, I use the node-based network to build a system that can generate all patterns from the data point. For example, for this patient with number 106, the whole file is about 30 minutes, which is equals to over 1.8 million milliseconds. If I know that there are 3 VT beats starting from 26 seconds, I could easily enter the time period and extract the patterns of those 3 beats and export them into my animation within Alembic file. It gives me a lot of flexibility to make a variety of ECG patterns. In the post-production phase, I used After Effects, Illustrator, and Red Giant to composite the whole scene and add in some additional special effects. So the whole animation series will be around 10 minutes long. So I'm not going to show all of them, but here is the 2-minute demo so that you can see.
In the realm of cardiac arrhythmias, ventricular tachycardia, VT, is the most ominous as it may cause hemodynamic collapse and lead to sudden death. To best appreciate the nature of DADs and EADs, it is critical to understand the nature of cellular regulation of intracellular calcium and how this process is mediated by the fundamental event of excitation-contraction coupling. In this angle, depolarization of the cell causes calcium channels, LTCC, within the cell membrane to open, triggering calcium influx into the cell, which results in calcium-induced calcium release from the intracellular calcium storage depot, the sarcoplasmic reticulum, the SR. Catecholamine-mediated VT, such as idiopathic outflow tract ventricular tachycardia, is considered a DAD-driven arrhythmia. It originates from the right and left ventricular outflow tracts and is triggered through beta-adrenergic stimulation, which leads to the activation of the stimulatory G protein GS-alpha and increased intracellular levels of CAMP and protein kinase A. EKA phosphorylates several important regulators of intracellular calcium, including the LTCC, the ryanodine receptor, and phospholambin, resulting in the extrusion of calcium from the cell and the influx of sodium ions. The latter results in a transient inward current consistent with a DAD. When the generation of DADs becomes an iterative process, it results in VT due to DAD-dependent triggered activity and spreads from a focal site to the whole heart.
So these 2 animations will be finished and published by the end of May. And before the official release, I will send it to my advisors for final comments and also collect feedback from my fellow classmates for the final retouch. You may access my portfolio under the motion media page to see the most updated ones later. Thank you so much for joining my thesis presentation. I would like to thank again my advisors and everyone here. Thank you.
Thank you, Harmony. That was very impressive, very, very cool animation. I have a quick question while other folks think about whether or not they want to raise their hand or have a question. Who did your narration?
The AI.
I wondered about that. It sounds very professional, but it's amazing that AI will do that. That you can get AI to do that kind of quality narration. And any quick questions? Again, anybody want to raise your hand or shout anything out?
Yeah, it's just Todd, Jim. These are all great presentations. I like this one, particularly prone to the heart stuff. Do you have any thoughts about taking it to the next level about the use of calcium channel blockers, other therapeutics for arrhythmia disorders, or particularly the one you're looking at, and layer in that animation?
Yeah, I think that's definitely something that I can do. I think there are a lot of molecular mechanisms that related to calcium ions because of the EC coupling process of the heart. So I think we can definitely make another animation to explain that process. But for now, because this animation is already very long to just explain EADs and DADs, so probably will be the next one.
Have to see if your
suggestion. Thank you.
I'd have to see if your content expert wants to take you on to take it to that next level.
Yeah.
But I know you already have a job lined up, so I guess that won't work. All right. So thank you, Harmony. Last but definitely not least, Brianna, you want to take over?
Yes.
Can everyone see this?
Here we go. If you can get it into presenter mode or full screen.
Can everyone see it?
Looks good to me.
Okay. It works. Okay.
Yeah, I can see it. I assume it means everybody else can.
Perfect. Okay. All right. Well, hi, everyone. My name is Brianna Hamilton, and this evening I'm excited to present to you my thesis presentation on caring for your heart during survivorship after pediatric cancer, a visual education guide on chemotherapy-induced cardiotoxicity. My scientific content advisor for this project is Dr. Brian Greffey, a pediatric oncologist at Children's Hospital, Colorado. To start, I'd just like to share a bit of background about myself. I was born in Colorado, but I grew up in Pennsylvania, where I found a love for creating art of the natural world and science. I have a background in cellular, molecular, and biomedical biology with a minor in visual art from Boise State University. And I'm currently working towards my Master of Fine Arts degree in medical illustration at the Rochester Institute of Technology. To begin, I would just like to share the inspiration and background behind my project. Why specifically the topic of chemotherapy-induced cardiotoxicity? Well, this project is largely inspired by my own experience with pediatric cancer. I was diagnosed at 22 months old with B-cell acute lymphoblastic leukemia, and I underwent 3 years of chemotherapy treatment at Children's Hospital, Colorado, where I was actually treated by Dr. Brian Greffey, who I shared as my scientific content advisor for this project. So this project is really personal to me. It's been a personal journey to reconnect with Dr. Greffey and collaborate with him for this project. Now, being a pediatric cancer survivor, I will need monitoring for the rest of my life, one of which is monitoring on my heart due to a type of chemotherapy drug I received called anthracyclines. To monitor my heart, I require regular echocardiogram screenings, which is a non-invasive way to monitor heart function. As I got older and I started to study science and understand science a little bit more, I really started to want to know the why behind all the tests and monitoring. As I mentioned, I have a background in biology, and yet I still felt in the dark about my own health, so I couldn't imagine what someone who doesn't have that background might feel like. That idea led to me wanting to help other survivors be better informed and help enable them to make educated decisions about their own medical care. I started looking into resources on cardiotoxicity and found that while there are some written ones that exist, few actually focus on education through visual media and tailored language for the general audience. That leads me to my project of creating a visual education guide. As I mentioned, I wanted to be able to help other survivors learn more about their own health for survivorship. As a result, this project was centered around these goals. Overall, I wanted to raise awareness of chemotherapy-induced cardiotoxicity, which is a long-term side effect of chemotherapy. By equipping survivors and their support systems, such as their families, with knowledge for advocacy, I want patients to be able to advocate for themselves and their health as they transition into survivorship. This project aims to serve as a resource to hopefully inspire questions and conversation that can foster communication between patients and their care team as they move into survivorship. And in doing so, I hope that they can be connected with any support that they may need for survivorship. And in speaking with Dr. Greffey for this project, we found that Children's Hospital Colorado actually offers a space where this project could reach the target audience of pediatric cancer survivors and their support systems. And that is through a unique clinic that the Children's Hospital Colorado offers called the Hope Survivorship Program. And that is a resource that connects patients with care teams to help patients transition from remission into survivorship and into adulthood. So the ultimate home of my project is at Children's Hospital Colorado in the Hope Clinic. I'd like to take a moment to go through an overview of the science behind chemotherapy-induced cardiotoxicity. So anthracyclines, this is a side effect caused by a specific type of chemotherapy drug called anthracyclines, such as doxorubicin or daunorubicin. These are chemotherapy drugs that are known to cause heart damage.
They do so by causing cellular damage in heart muscle cells called cardiomyocytes, which you can see in this diagram I've illustrated here. The damage weakens a part of the heart called the left ventricle, which is the part of the heart that's responsible for pumping blood throughout the rest of the body. So damage to this area of the heart results in a heart that cannot pump blood as effectively as a healthy heart could. Now, these drugs enact damage through a series of molecular mechanisms that are outlined in the diagram you see here. And these mechanisms include the generation of reactive oxygen species, oxidative stress buildup, calcium homeostasis disruption, all of which leads to the activation of apoptosis or programmed cell death in cardiomyocytes. These drugs also enact damage through inserting themselves or intercalation into DNA, and they inhibit an important enzyme involved in DNA replication called topoisomerase 2 beta. Now, these drugs kind of behave as a double-edged sword. All of these mechanisms are what the drugs are intended to do to fight off cancer. So these mechanisms induce cell death in malignant cells and kill cancer cells. Unfortunately, these same actions are what can lead to cardiotoxicity through harming healthy cells in the heart and can ultimately lead to heart damage and heart failure in serious cases. But I'd like to share a disclaimer, though, that not every patient who receives anthracyclines will experience cardiotoxicity. The risk of developing this side effect is largely based on the total dose of anthracyclines that a patient received during the length of their treatment, and if that treatment was combined with chest radiation. The risk is increased if radiation was involved in treatment. Healthcare professionals can evaluate a patient's risk through resources such as the comprehensive guidelines published by the Children's Oncology Group. These guidelines are a nearly 400-page document on several different types of chemotherapy side effects, not just anthracyclines, and they are designed to help provide ongoing care to childhood cancer survivors. And although the information in those guidelines is valuable to survivors, the only version currently available is targeted to healthcare professionals. So this means that it is very text-heavy and filled with scientific terminology that would not be clear to the general audience. So this is where creating a more visual resource that has tailored language comes into play. So to create visuals for this project, I started with background research by reviewing histological images of cardiomyocytes. I watched heart dissection videos. I reviewed current medical illustrations of the heart, such as those created by Frank Netter. And I looked at techniques or iconography of monitoring techniques because I mentioned on the guide an echocardiogram. So I thought that it was important to include an imagery of an echocardiogram in case patients had never seen that machine before. So I went through this process of creating the 2D illustration assets for the guide. The research process, I created sketches, then went through revisions, and created the final illustrations in Adobe Photoshop and Adobe Illustrator. An additional element to the guide that I included was the inclusion of 3D interactive media. As I previously shared, cardiotoxicity is a complicated cellular molecular process, which is way more information than patients actually need to know, but I wanted to help enhance their understanding of the anatomy that is mentioned on the guide and ultimately increase engagement with the guide itself. So cardiomyocytes, or heart muscle cells, are mentioned on the guide. So I created this stylized 3D model of a cardiomyocyte in Autodesk Maya. And it is accessible by scanning a QR code found on the guide that will take patients directly to the colorized model that you see here on the screen. So I paid it when I was creating the model of the cardiomyocyte, I paid close attention to keeping the geometry of it very simple and stylized. Different parts of the model were assigned a material shader to keep it grayscale while it was in Maya. And then the model itself was exported as an FBX file and uploaded to another software called Sketchfab. Sketchfab allows the colors to be applied within its own software, and it's an internet source that hosts the model and allows for accessibility and interactivity. And once patients scan the QR code, it takes them to the model, and they can interact with the model in the way that you see here in this video. It allows the model to be spun around. And then I've also included models of organelles that are involved that are part of cardiomyocytes. Now, organelles such as the mitochondria, sarcoplasmic reticulum, and intercalated discs are not mentioned on the guide itself, but they are important in the overall mechanisms of cardiotoxicity. So I thought that it was important to include them in case patients want to learn more, so then they will have already seen those aspects once they interact with the guide. When I was creating the guide itself, there were a couple design considerations that I had to keep in mind. As I previously shared, chemotherapy-induced cardiotoxicity is a complicated process that involves many cellular molecular actions, but my audience for this project is pediatric and their families, so it was imperative that the language be tailored to that audience, and it was tailored to an 8th grade reading level, as was suggested by Dr. Gruffy and other care team members of the Hope Clinic. So here you can see the breakdown of the process I went through to tailor that language. So first, which was way too complex, was information right from the peer-reviewed resources that I was reading. Less complex was me digesting that information and kind of rewriting it in my own way as I understood it. And then finally, I looked at tailoring the information to include only the most essential terminology that patients would need to be aware of for this side effect. Additionally, because there is quite a bit of text, even if it is simplified and clear text, there is still scientific text on the guide itself. So I wanted to include some fun elements to help break up that scientific text. That includes like the heart giving itself a hug, the loopy arrows, or the little call outs to certain phrases on the guide. It just helps draws attention to certain elements on the guide itself. And this is a mock-up of the final guide. You can see how some of the elements I've mentioned have come together with the 2D illustrations, those fun elements, and then the large QR code that patients would scan that takes them to that interactive model of the cardiomyocyte that I previously mentioned. And this is the full layout of the guide itself. The guide itself is a trifold pamphlet that consists of 6 panels, 3 outer and 3 inner panels. And on the guide, patients can expect to find some of the scientific background on cardiotoxicity, where it mentions cardiomyocytes and the left ventricle. It'll share who is at risk for developing cardiotoxicity, again, though it'll give the disclaimer of that it is dose dependent. It'll share how patients can care for their heart without pharmacological intervention. It'll share how they can engage with their care teams through asking questions. And then there is a section on long-term follow-up care through echocardiogram monitoring. Now, next step for this project actually include implementation at the Hope Clinic in Children's Hospital, Colorado, in Aurora, Colorado. And the guides will be made available to patients who received anthracyclines. Now, because anthracyclines are used to treat certain cancers, only patients who received anthracyclines as part of their treatment regimen will need access to this information. And then after interacting with the guide, there is an option for viewers to provide feedback through a survey that's hosted through SurveyMonkey that allows me to gauge if patients thought that information was made more accessible through artwork and visual media. And then working on this project, it opened up kind of some thoughts and some opportunities to create additional visual guides on other long-term side effects that are described by written resources, such as those from the Children's Oncology Group. Cardiovascular risks or effects, such as cardiotoxicity, are unfortunately just one type of side effect of chemotherapy. There are a lot of others that patients experience, such as dental health issues, skin health issues, and bone health issues that patients could benefit from a visual guide on. I just want to take a moment to acknowledge everyone who supported this project in my graduate school journey. Of course, to Dr. Gruffy, the medical illustration faculty, my incredible cohort who you have heard present here tonight, my family and friends and loved ones, and not only with my education and thesis, but a huge thank you to everyone who supported me with my own experience with pediatric cancer. I would not be here without you. And I want to give thank you to the Hope Survivorship Program and Children's Hospital Colorado for providing a home to my project to help reach other pediatric cancer survivors. This concludes my time. I thank you for your time and for listening to my project on caring for your heart during survivorship after pediatric cancer. Thank you.
Thank you so much, Brianna. Excellent presentation. Beautiful work. Any questions?
Yeah, Jim,
Todd,
Todd.
I thought you might have one.
Yeah, no, well, first of all, kudos to everybody and you and the program. Outstanding. Question, did you come across when you talk about the dose? I know in the past it was like 600 milligrams per kilogram, but did you come across any specific range associated with the tendency then to be predisposed for cardiotoxic effects?
Yes, currently by the Children's Oncology Group, if it's above 250, and especially once you get into the 300 range, there is the great increased risk of developing this side effect.
Thanks. Yeah, I was curious because I know in adults, I think it's more about twice that. So that's really important. And glad to see the shout out for the Colorado Children's Hospital. I have a colleague that I trained with, Doug Graham there, and he's a pediatric cancer doc. So cool.
Cool.
Yeah, I had a question too about the science. I know, I think you and I have talked about this before, Brianna, but I couldn't remember the name of the exact drug that I was thinking of. About 20 years ago, I did an illustration for an oncologist specifically about cardiotoxicity with doxorubicin specifically. And at that time, I believe it was an experimental drug, dexrazoxane, that could be administered simultaneously with the doxorubicin to have a sort of cardioprotective effect. Is that something you came across, or I mean, this is I did this illustration long enough ago that may have been when you were undergoing treatment. So I'm curious if you're familiar with that.
Yes, that was actually a drug that Dr. Griffin and I talked about that they do, it's been FDA approved. So that is the only drug that's been FDA approved that they give in conjunction with the actual chemotherapy drugs. It was not approved when I was going through treatment, is my understanding. So it was not available for me. But I was not at the high risk of it. But yeah, my understanding is currently for children that are high risk of developing the side effect. Yes, it's the dexrazoxane that they give.
Okay, great. Thanks.
Mhm.
Any other questions specifically for Brianna?
Hey, great presentation, Brianna. I loved it. And I had a question for you. How did it become known that the effect of chemotherapy had on the heart? How did it become known, and what did doctors do before they had the treatment for the heart?
Yeah, my understanding is that they've known since the 70s that chemotherapy has effects on the heart. Unfortunately, back then they didn't know the mechanisms. Many of the molecular mechanisms I talked about, they were implicated in the process, but like the inhibition of topoisomerase 2 beta was, as of 2020, was fully implicated in the process. So back then in the 70s, the survival rates were a lot less. Unfortunately, they were around 50% compared to today, they're upwards of the 90%. And this is also a side effect that more patients are likely to experience up to 20 years after receiving treatment. So it's not something that they usually experience early on. So unfortunately, back then, my understanding is that there wasn't a whole lot they were doing or could do until patients showed effects of it.
Awesome. Thank you.